Hello and welcome to the presentation on chapters 9 and 11 on immunological tolerance and disorders caused by immune responses. So, so the things that are going to be kind of the takeaways here or that are going to be discussed are how does the immune system maintain unresponsive to self-antigens? What are the factors that may contribute to the loss of self-tolerance and develop autoimmunity? And what are the mechanisms of different types of sensitivity reactions, hypersensitivity reactions? I'll be covering kind of the first two questions, and then Brianda will get covered the last question. So here's a picture or diagram of central tolerance and peripheral tolerance against self-antigens, or recognizing those self-antigens. So the body needs to be tolerant to self-antigens, or serious side effects of autoimmunity can ensue. These self-antigens need to be part of our body in order to maintain homeostasis. Therefore, there are multiple tolerance mechanisms in order for this to happen. One of those mechanisms is central tolerance. This involves immature lymphocytes that recognize self-antigens to go through certain changes in their lymphocytes. Central tolerance involves lymphoid organs, the thymus, and bone marrow. The results that can occur are apoptosis, change in receptor editing for the B cells, and development of regulatory T lymphocytes for CD4 plus T cells. Another mechanism is peripheral tolerance. This involves mature lymphocytes that recognize self-antigens. The outcomes that can come from this are anergy, apoptosis, which is deletion, and suppression. To kind of think of it, central tolerance is just more involved in changing immature lymphocytes because they have not matured yet, and they can still kind of change their receptors and kind of change their overall cell. But for peripheral tolerance, they're kind of more involved with kind of getting rid of that cell because it is matured. So going on to factors that affect the body and getting autoimmune diseases, we kind of covered how to prevent those self-antigens from harming us. But now we're going to be talking about what are the two things that allow autoimmunity to occur. These would be genetic susceptibility and environmental stimuli. Genetic susceptibility will affect the lymphocytes, genes, and find the loci that maintain self-tolerance and alter it. The main loci that will mostly be affected here are the MHC genes because they are so diverse and go through so many mutations. Inbreeding of animals or humans will cause more likelihood of these MHC genes to be altered. And sometimes it's not going to be the MHC that's going to be altered, but maybe it will just be a single gene within that genome that alters its function. So it will cause autoimmunity and have more mutations. The environmental stimuli will allow for autoimmunity to occur from inflammatory processes and activation of self-reactive T cells. Kind of the two general ways this can occur is through self-tolerance, anergy or deletion, and autoimmunity. Now Brianda will be talking on her part. Thank you.
Mechanisms of different types of hypersensitivity reactions. But first, what are hypersensitivity reactions? We understand that the immune system is normally supposed to aid and protect against microbes and foreign antigens and keep us healthy, but sometimes these reactions can contribute to tissue injury as well as disease. Now, let's get into type 1 hypersensitivity, also known as immediate hypersensitivity. This hypersensitivity reaction is mediated by immunoglobulin E antibody against environmental antigens such as pollen. This sequence of mechanisms starts with activation of the T helper 2 and interleukin 4 secreting follicular helper T cells. It then stimulates production of the IgE in response to the antigen, and then 3, it binds to the FC2 mast cells and release of mediators that induce the allergic symptoms. The effects are allergies like hay fever, food allergies, asthma, and anaphylaxis. Moving on to type 2 hypersensitivity, it is mediated by antibodies, most likely which is IgG, when binding to their target antigens in different tissues. The antibodies are usually autoantibodies that result from failure of self-tolerance, spoken about by David. Antibodies directed against self and extracellular matrix deposit in whatever tissue that indicates the relevant target antigen. And it causes injury by inflammation, opsonization, and phagocytosis with the macrophages and destruction of cells and or limiting their function. Type 3 hypersensitivity antibodies can form immune complexes that deposit in excess in the blood vessels. This can result in widespread vasculitis in organs such as the kidneys and joints. Complexes with positively charged antigen are particularly harmful as they bind more easily to negatively charged components of the blood vessels and kidney glomeruli. The Fc regions of antibodies can activate complement and bind to Fc receptors on neutrophils. This causes the release of harmful substances such as protease and reactive oxygens, which can exacerbate inflammation and tissue damage. An example of this is rheumatoid arthritis. Finally, we have type 4 hypersensitivity, which is mediated by T cells usually due to autoimmunity and is amplified with constant responsiveness to microbes and other environmental antigens. Due to the inflammation caused by the cytokines developed by CD4, Th1, and cells of or killing of host cells by the CD8 cytotoxic lymphocytes causes all these symptoms. The typical reaction by T cells is delayed type hypersensitivity, which occurs 24 to 48 hours after an individual was previously exposed with an antigen and is then re-exposed. An example of this is the tuberculin test, also dermatitis with poison ivy, and sometimes medications can have this effect.